Let's take a look at these mutations now. Okay, so mutation is essentially a mistake during DNA replication. So what does that mean? Well, if you go up here, DNA replication we know uses this semi-conservative model. So essentially you have your original parent strand. It splits into half. Okay, these blue strands are the original, right? And you can see A, T, C, G, cars go into the garage and apples grow in trees. They split apart, and then these free nucleotides go bloop, 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 and they attach. And then what you get in the end is two identical daughter strands. This is identical to the original parent, except for this is the new side, and this is the original side right there. This is the original side right there, and this is the two new, right? So half new, half old. Now what can happen is in this process where these free-floating nucleotides kind of attach, sometimes the wrong one will sit there and it gets stuck there and it doesn't belong. So then here's an example right here of the original parent strand, and then this is the new complementary strand. So that'd be the gray side on that picture. And you can see apples grow on trees, trees, apples, trees, trees. That's not good. Okay, and then cars in the garage, apples, trees, cars in the garage, apples, trees, cars in the garage, or garage, however you want to say it. So this one here, there's a mistake. This is the original strand, so that's not the mistake, it's this one right there. So this should be an A. It should be an A. Now, let's say you're using the DNA codon, or DNA triplet list, okay, and TAT was going to be one of the amino acid triplets, and then here's another triplet right there, and there's the other triplet right there. So what you would find is that this one here might be a different amino acid. These are the original. That's where the mutation occurs. So there's a mistake in DNA replication that results in a different triplet, which then results in a different amino acid. Now you can have a mutation that actually doesn't change the amino acid. The triplet's different, but the amino acid is the same, and that's because, remember, in this list we have 20 amino acids, but we have 64 triplets, and that's called redundancy. So we have more than one code for each amino acid. This one has 6. My goodness. That's all. So you could have mistakes like TTA could change to a T, this A could change to a G, still be leucine, right? Get the idea? Okay, so mutations. So sometimes those are called silent mutations. If they give you the same amino acid, it's silent. You don't even notice. So let's take a look at the rest of this. So one or more nitrogen base is changed in a gene. Not all mutations will change the amino acid. Why? Because of the redundancy. Because amino acids, there's many triplets for one amino acid. It's technically, this is called redundancy. Okay, that's why. So here we go. We got our original strand, then we have the mutated. So we have ATG, ATG, CGA, CGA, TTC, TTT. So you have to spot the mutation. It's that right there. And then we have TGA, TGA. So here's the original amino acid chain. So the only one that's really been changed here is the third amino acid. So it's this one right there. So the rest of this, we can go like this. We can go, that's going to be methionine. That's going to be arginine. And then what's TTT? So we go up to our list. Okay. Look for T, T, G, leucine. TTT, phenylalanine. Okay, so let's put that as PHE. Hopefully that'll work. And that's going to be, look at that. And then this other one is the original. That's going to be a stop codon. So that's the same, which means that's a silent mutation. It doesn't change anything, really. It's a different code, but it's the same amino acid. Okay, let's try this next one here. So the original and the mutated. Okay, We got ATG, ATG, CGA, CGA, TTC, TAG. Okay, that's the mutation. So it looks like the A and the T. The A has been changed. Oh, look at that. And the G. Okay, so 2 of them have been changed. And TGA, TGA. All right, so this is going to be different. Just that one. So this is the one that's been mutated. That's once again phenylalanine. Okay, so we're going to have methionine. We're going to have arginine. A lot of creativity here in these questions. TAG, what the hell is that? What the heck is that? Sorry. Nope. AG. It's a stop codon. Okay, so it's going to be a stop. Oh my goodness. So then, essentially, instead of having 3 amino acids, we only have 2. That's very different. Okay, that's going to be a bad mutation, most likely. That is not going to be silent. This one here, ATG, ATG, GCG, CGA. Oh man. Okay, so that's mutation, mutation, mutation. Here's mutation there, the A. There we go. The G, TGA, and that's it. Okay, so this one here is going to be mutated, and this one here is mutated. The rest are the same. It's going to be methionine. CGA is what? CGA? CGA is arginine, ARG. Okay, so that's not the same. And then TAG, it was phenylalanine. Let's see what it is now. TAG, sounds familiar. TAG stop, so TAG stop. All right, so that's not good. So once again, you're losing, right? This is only 2 amino acids, and there was 3. So that's going to be a bad mutation. Okay, so hopefully that makes sense. A mutation is a mistake that we have during DNA replication where the wrong nucleotide matches up. So instead of following the cars go with the garage, CG, and apples grow on trees, AT, you get a mess up. And it can lead sometimes to a different amino acid, but because there's redundancy, multiple triplets for the same amino acid, sometimes it makes no change, and that'd be called silent. All right, hopefully that is